Understanding GWAS, Identifying and Analyzing Genetic Variants Linked to Diseases. Title: Understanding GWAS, Identifying and Analyzing Genetic Variants Linked to Diseases. Introduction, 100 words. Genome-Wide Association Studies, GWAS, have revolutionized our understanding of the genetic underpinnings of diseases. By analyzing vast amounts of genetic data from thousands of individuals, GWAS enables researchers to identify genetic variants associated with the development of diseases. This article will delve into the fascinating world of GWAS, exploring its methodologies, benefits, and limitations. We will explore how these studies have advanced our knowledge of diseases, laying the groundwork for personalized medicine and the potential for targeted treatments. Overview of GWAS, 200 words. Genome-Wide Association Studies are observational studies conducted on large populations to identify associations between genetic variants and diseases or traits. By comparing the genomes of ALS with and without the condition of interest, researchers can pinpoint genetic markers associated with specific diseases. GWAS analyze millions of single nucleotide polymorphisms, SNPs, which represent common genetic variations across a population. These SNPs can reside in coding regions or non-coding regions of the genome, impacting gene expression or protein function. 2. Methodologies and techniques, 400 words. GWAS follows a systematic process that involves several key steps. Firstly, scientists collect DNA samples from thousands or even millions of individuals, including both patients and controls, to establish a robust dataset for analysis. Once the DNA samples are obtained, researchers harness cutting-edge genotyping technologies to identify millions of SNPs across the genome. High-throughput genotyping arrays, such as microarrays or Next-generation sequencing techniques, facilitate the simultaneous detection of thousands of SNPs. The collected data is then subjected to statistical analysis. To reduce the risk of false positives due to multiple comparisons, a significance threshold known as the genome-wide significance level is established. This threshold takes into account the number of SNPs tested across the genome and adjusts the statistical threshold accordingly. 3. By unraveling genetic variants associated with complex diseases like diabetes, cancer, and Alzheimer's, GWAS have paved the way for personalized medicine. Additionally, GWAS have uncovered shared genetic components among seemingly distinct diseases, leading to the discovery of shared pathways and molecular mechanisms. This has facilitated the repurposing of drugs originally developed for one condition to be used in the treatment of another. GWAS results have not only advanced our understanding of diseases but also accelerated drug development. Furthermore, GWAS have challenged traditional notions surrounding disease classification. Rather than grouping diseases based on symptoms alone, GWAS has provided evidence for a molecularly guided classification. This enables the identification of subtypes within diseases, allowing for targeted treatments specific to an individual's genetic profile. 4. Limitations and challenges, 400 words. Despite its remarkable success, GWAS has certain limitations and challenges. First, GWAS often identifies common genetic variants associated with diseases, which usually have very small individual FECT sizes. These variants may explain only a fraction of the overall disease risk, leaving a substantial portion unexplained. Additionally, GWAS primarily focuses on the identification of associations, rather than elucidating the underlying biological mechanisms linking genetic variants to diseases. This poses a challenge in understanding the functional consequences of identified variants and limits the translation of GWAS results into clinical practice. B. Emerging trends in GWAS, 300 words. As technology continues to advance, new trends are emerging in the field of GWAS. One such trend is the integration of multi-omics data. Another emerging trend is the use of large-scale biobanks. Biobanks store biological samples and associated health information from large populations, providing a valuable resource for GWAS. These biobanks allow researchers to access large cohorts of individuals, increasing statistical power and enabling the discovery of novel genetic variants. Furthermore, there is a growing emphasis on functional genomics in GWAS. Functional genomics investigates how genetic variations impact gene expression and protein function. Integrating functional genomics data with GWAS results can help pinpoint causal variants and shed light on the biological mechanisms involved in disease development. In recent years, there has also been a focus on fine mapping studies. Fine mapping aims to identify the specific genetic variants within a genomic region that are responsible for the observed associa. Tie-in with a disease. This approach helps narrow down the potential causative variants and improve our understanding of the functional implications. Additionally, with the advent of single-cell genomics, researchers are able to study genetic variations at a cellular level. This enables the identification of cell-specific genetic effects, providing insights into the heterogeneity of diseases and potential therapeutic targets. Moreover, the integration of GWAS data with electronic health records, EHRs, is becoming increasingly important. EHRs contain comprehensive medical information for large patient populations. By Linky. In GWAS findings with EHR data, researchers can investigate the clinical implications of identified genetic variants and develop more accurate risk prediction models. One key concern is the protection of participants' privacy and the responsible use and storage of their genetic data. This includes implementing robust data security protocols and adhering to strict informed consent processes that clearly outline how the data will be used and protected. Another ethical consideration relates to the potential for stigmatization and discrimination based on genetic findings. As GWAS identifies genetic variants associated with diseases, there is a risk that individuals may face discrimination in areas such as employment, insurance, and relationships. It is essential to develop policies and enforce regulations that prevent genetic discrimination and protect individuals from any negative consequences resulting from their genetic information. Furthermore, the representation and diversity of study populations in GWAS are important ethical considerations. Historically, GWAS studies have predominantly focused on populations of Europeans, leading to a lack of diversity in genetic research. This can result in disparities in understanding disease susceptibility and treatment efficacy among different populations. Efforts should be to include diverse populations in genetic studies to ensure the equitable distribution of the benefits of GWAS research and to avoid perpetuating existing health disparities. Transparency in GWAS research is another ethical concern. Researchers should disclose potential conflicts of interest and be transparent about the funding sources to maintain integrity and trust in the scientific community. Open access to GWAS data can also promote collaboration and ensure that the findings are accessible to the wider scientific community for further analysis and validation. Continued engagement and communication with participants is crucial to maintain trust and ethical standards in GWAS. Participants should be kept informed about the progress and outcomes of the research. And should have the opportunity to provide input and feedback on how their data are used. One of the key areas of focus is the integration of GWAS data with functional genomics and epigenomics. This integration can help uncover the causality between genetic variants and diseases, providing a more comprehensive understanding of the biology behind these associations. Another promising direction is the inclusion of multi-ethnic populations in GWAS studies. Historically, the majority of GWAS have been conducted on populations of European ancestry, which may not captain the full spectrum of genetic variation across diverse populations. Furthermore, the application of machine learning and artificial intelligence (AI) algorithms is expected to enhance the analysis and interpretation of GWAS data. These technologies can help identify complex interactions between genetic variants and environmental factors, leading to more accurate risk prediction models and personalized treatment approaches. In addition, there is a growing interest in investigating the role of rare genetic variants in disease susceptibility. While GWAS primarily focuses on common variants, rare variants with larger effect